Okay, so hello everyone. Welcome to our today's Zoom seminar. So it's absolutely my pleasure to welcome our today's speaker from Duke University, Dr. Diego Bohórquez. He's currently Associate Professor of Medicine and Neurology in Duke University. So a very, very brief introduction about Diego. So Diego got complete his PhD training in nutrition in North Carolina State University. And then
Mhm.
from 2010, he started his postdoc training in Duke University in department in Duke University. And then from 2015, he started his independent research program as assistant professor in Department of Medicine and Neurobiology in Duke University. So 2021, Diego was promoted as associate professor also in Department of Medicine, both the same time in Department of Pathology and Neurobiology. So Diego is a leading neuroscience scientist of the lab of gut-brain neurobiology at Duke University. So his lab is really have a long-term vision to heal the brain from the gut, and his research team is really doing some groundbreaking work about our understanding of how gut sensations influence our behavior and choices. So, but more than that, Diego's research also all about creating innovative tools to advance the field. His team has developed a flexible fiber optical for wireless optogenetic control of gut neuropods in living animals, allowing for real-time manipulation of gut-brain communication. This really pioneer work paved the way for novel therapeutic innovations to heal the brain by acting on the specific cell from the gut. So with really a lot of refreshing and groundbreaking work, Diego got numerous awards and honors. I'm not gonna list them all, but including NIH New Innovator Awards. 2020, he got Cavalier Fellowship, and then 2022, he's become a faculty fellow of the Jewish National Fund. So besides all these, the background and introduction, I've been fortunately to hear Diego's talk for a couple of times. This is really entertaining. I think everybody online will have really great time tonight. I can guarantee you. So with all the pleasure, thank you, Diego, for doing this for us. The floor is yours. We are very much looking forward to your talk.
Thank you, Chuan. Can everybody hear me? Can you hear me?
Yes.
Okay, perfect. Another checkpoint in there. You should be able to, let's see. Can you hear a little bit of sound in there?
I can hear the sounds. Yes. Yes,
Yep,
music.
beautiful.
See?
All righty.
Well, the show is on.
Thank you so much, everyone. It is my absolute pleasure to be here. I am always honored to be invited to the stage because I think that every time that I come to a stage, my world expands. And when I say that my world expands, I wanted to bring up this book from Ed Young. It's called An Immense World, which I just listened through it twice last week. Twice because I just couldn't get enough the first time. And I highly recommend for you to go through it. And the way that Ed opens up the book, he talks about this word that I was not aware of. It's called Umwelt. Umwelt. So this word Umwelt was coined by Jacob von Wuxkull to say that essentially we are what we can, our senses can perceive. However, that's not everything that it can be perceived, as you guys can probably attest, right? So essentially we live in this sensory bubble. Like 15 minutes ago, before you and I met through here online, your sensory bubble was very different. And right now, my sensory bubble has expanded. And this hopefully starts to expand our idea that we only have 5 senses, you know. The canonical ones for sound, smell, taste, touch, sight. And while we began starting to understand the cellular basis of these canonical senses since the 1950s, in the gut, which is the only organ still exposed to the surface that goes through the entire body. You know, the nose doesn't do that, the reproductive tract doesn't do that, the ear doesn't do that because it goes in and then comes back out. But the gut goes through the entire system of the body. And it is not internal, it's actually still external, because if you eat a marble, I assure you that the marble will not become part of you. But if you eat food, that food will become you and you will become that food, right? So when you look at here, at the way that we thought about the gut, not long ago, 10, 15 years ago, it was rather simplistic. So what happens when we eat food? When we have a piece of chocolate, we chew on that piece of chocolate to bring it down into pieces, and that forms into a chyme. And that chyme is brought down into the stomach, where it's bathed with some hydrochloric acid, some pepsin, some rennin. And then the gatekeeper, Mr. Pylorus, lets the chyme flow into the intestine. And it is in the intestine where the magic happens, because in talking with even some philosophers, that is where literally food becomes us and we become food, because if we are what we eat, this is the point where food is internalized. This is the point where the gut knows how to extract the proteins, the fats, the sugars to continue to build our body. It is also the place where lots of microorganisms live in, that helps us to utilize that food, but also we help them to have a community. And my fascination for the gut began in grad school. Here you can see some scanning electron micrographs in which you have a single volus that when it loses its tip, you can see how beautifully all of the cells are arranged around this core. And that core is the one that has all of the immune cells, the muscle cells, the lacteal, the blood vessels, and certainly the nerves too. But up until like 2007, we had a very simplistic view of how it is that nutrients, when they enter into the intestine, they are recognized by the intestinal epithelium. And the idea was that when the nutrients enter into the intestine, they will be digested, and then they will stimulate these so-called enteroendocrine cells to release hormones. That term, enteroendocrine, was coined in 1938. Frederick Feiter at that time was a pioneer who had figured out that the gut has this diffuse endocrine organ, meaning that as the chyme went through the intestine, there was a lot of hormones that were released. So he coined this term enteroendocrine for the cells that were releasing those hormones. But in 2008, there was a revolution that started to happen. Professor Frank Raymond and Fiona Gribble at the University of Cambridge were the first ones to utilize a GFP reporter mice to be able to study these cells. So these cells had been first observed by Rudolf Heidemann in 1875. And throughout all of this time, we have been studying them through indirect mechanisms and indirect means. But when the transgenic reporter GFP mice came in, all of a sudden, not only was possible to identify them, but it was possible to isolate them, take them out, sequence them, and start to understand how it is that not only they sense, but also how is it that they communicate what they have sensed. And this is around the time that I got into the picture. And I remember talking with a friend and mentor at the time, the late and legendary Oliver Smithis, who won the Nobel Prize in 2007. And I remember telling him about my excitement of having seen these cells. And Oliver said, so you have been bitten by the bug. He said, like, you are, that bug called science will never leave your body now, he said, because you will always be curious and you will always be fascinated by the next question. So the next question was putting these tissues from these mice underneath the microscope. And as you can see here, very quickly, we started to see something different that we have observed under the microscope. So one of the things that became evident is that some of these cells, especially those cells in the distal part of intestine, they had this process underneath the epithelial layer that eventually we gave it a name and we call it neuropod. Because the cells were called enteroendocrine or gut cells that release a hormone, I thought immediately the cells must be connecting to the vasculature. Wrong. As you can see, the cells are not connecting to the vasculature. They are actually very separate from the vasculature. So eventually my mentor said, like, Diego, you need to publish something because otherwise we're gonna run out of money and we will run out of funds. So one thing that I wanna highlight is that this was my first manuscript. So I'm very thankful for that opportunity to Roger, because with this manuscript, I learned to at least put together, I don't know that I learned to do science, but at least to put together a report of science. And one of the things that I highlight here is that submitted 14 September 2010, accepted 19 October 2010. Those things don't happen anymore, so I always highlight that. So for the next 3 years, with this, I was able to secure a fellowship. And then for the next 3 years, I had the opportunity to begin exploring and understanding not only the cells, but also their environment, their Umwelt, so to speak. So here you have a reconstruction of 510 micrographs consecutively collated that had been taken with a scanning electron microscopy and manually reconstructed. And you can see these neuropod cells in here. So in green, you have that neuropod cell. And then underneath, you have that subepithelial myofibroblast that form these holes, these fenestrations. And then one of the things that we found is that underneath there were cells that were physically interacting with these neuropod cells. And obviously this was an eye-opener, and somebody at the end of the conference asked me, said like, hey Diego, does the microscope also puts music on it? And I said like, no, when you start to you spend like 600 hours looking at this data, then you start to hear some things, and I wanted to share that with you guys. And it really opened my mind, because then we immediately started to put some glial-derived neurotropic factors to label the glia. And you can see here in immunohistochemistry how beautifully these glia come in and come into approximation with those neuropods. And not only they come into approximation with those neuropods, but if we take a cholecystokinin label cells in these intestinal organoids, and now all of a sudden we put some of these nerve growth factors, then the feet, these neuropods, start to be extended, as you see here. And if you put NGF, you see an extension of the neuropod. If you put artemin, which is a branching factor in axons, then you see like many different feet. And we published that work in there. And I highlighted because it really needs some brave souls to go in there and expand on that work, because I do believe that it has tremendous potential for neuroimmune interactions. So we came back to the question of what will be the cells that these neuropods are connecting to. And here you can see a labeling with VIP, vasoactive intestinal peptide nerve fibers. And we started to get some clues. And as you can see here, they do not connect. And they do not connect because these VIP labeled neurons are actually motor neurons. So if you think about it, a sensory neuroepithelial cell shouldn't be connecting to a motor neuron, because a motor neuron is classically known as an effector. But if you put a marker of sensor neurons, PGP 9.5, then all of a sudden you start to see a different kind of label. In fact, depending on the marker that you will use, you will have anywhere from 1/2 to 1/3 to 2/3 of the cells that will be in close contact with nerve fibers. And if you go carefully through the literature, there has been some examples in which, as you can see here, these are serotonin level cells in the rat epithelium, that they are actually physically connected even after digestion. So 1 in 5 of those is still connected to the nerve fiber. Here you can see another example. But the clear question was, are these cells truly connecting, or are they simply in close apposition or in contact with nerve fibers? So at some point we ran this experiment in which we took out a sensory neuron from the brain. And the first time we did it with trigeminal ganglia neurons. And we did it with trigeminal ganglia neurons not because we didn't know that the trigeminal ganglia does not innervate the viscera, but because the trigeminal ganglia is rather easy to access for sensory neurons. So when we put it in contact in culture with these neuropod cells, you're gonna see what happens. So on the left-hand side, you have one of these green neuropod cells labeled by CCK, and on the right-hand side, you have one sensory neuron. And take a look what happens at hour 12. So not only those cells are able to recognize each other, but they are able to recapitulate the circuit, and then all of a sudden their umwelt has completely expanded, because now the neuron is able to grow this axon, but also has the sensory input of this neuropod cell, right? And of course, there are so many things in there, that we don't understand. However, it really began to become a platform for us to begin to understand how these neuroepithelial interactions happen. And one of the beautiful things here is that if we put an organoid, an intestinal organoid, next to a nodose ganglia neuron, as you see here, the nodose ganglia invariably extends some of its axons into the epithelial layer of the organoid. And then if we use a monosynaptic rabies virus, we can label those neuroepithelial cells, as you can see here. We have a CCK GFP organoid, and then we put this tomato rabies or this cherry rabies, there is overlap. But here is the fascinating thing. We can run a monosynaptic tracing from a vagal nodose ganglia neuron into an organoid. So if we have a wild type vagal nodose neuron and we put an MV rabies that lacks the glycoprotein to be able to jump one synapse, the rabies gets into the neuron but is not able to jump the synapse. However, if we bathe that vagal nodose neuron with the RAPG complement, and now we put the rabies inside of the neuron first, you can see that it labels one of these one of these neuropod cells, meaning that the rabies virus was able to jump specifically in one of these neuroepithelial cells of the organoid. And then it quickly became evident that these vagal nodose neurons themselves alone cannot recognize a stimuli. If we put glucose in there, it does not elicit any of excitatory potentials. However, if we co-culture them with these neuropods, now all of a sudden, not only you have these excitatory postsynaptic potentials, but you also have action potentials. And this is literally, as many of you know, the basis of sensory transduction, the transduction of a chemical into an electrical pulse that ultimately is guided through a circuit, perhaps to drive behavior, right? And we published some of that work in 2015. And then it became clear that for a while, it became a little bit daunting because we didn't know that we were the only ones working in this area. However, in 2017, Professor David Julius was glad to see that not only he had expanded on this work, but also shown that the serotonin cells in the colon, they also seem to form the synaptic-like contacts and then send in signals onto sensory neurons. So we began tracing the circuitry, because as you all may know, depending on what neuron the sensory cell is connected, then the effect will be very different, right? So in a leap of faith experiment, we put some of these rabies into the lumen of the colon of the mouse. And 7 days later, you can see here that there's a specific population of these neuroepithelial cells, neuropods, that are labeled by the rabies. And then if we create a transgenic mouse in which it expresses this RAB-G-GFP, now all of a sudden, when we put the rabies will infect this epithelial cell, will turn it green, will recombine with the RAB-G, and then jump one synapse and stop there. And this is what we saw after 2 or 3 years of work, that it's literally the place where the gut meets the brain, because what is in red is a cell labeled with PYY, and what is in green is the GFP from the rabies virus. And upstream of that, you see 3 vagal nodose ganglia neurons that had been labeled by the rabies that was put in the lumen of the colon. And then obviously above the vagal nodose ganglia neurons, they end in the nucleus tractus solitarius. So these experiments brought up 2 consequences immediately. One is that the gut, like the tongue, like the nose, like the skin, also has the anatomical basis to transduce stimuli very rapidly to the brain. And the second one is that inadvertently, by using rabies, we have bumped into a potential path for pathogens to crawl up the brain. And I highlight that because there's quite a bit of evidence that neurodegenerative diseases may begin in the periphery. However, the circuitry is still a little bit unclear, although there is quite a bit of evidence that it may begin in the lumen of the peripheral organs. And some of that work, we had published it over here. So now finally, if we have all of these circuitry and all of these elements, what will that be good for? And here's one of my favorite initial lines in a paper. So in 1973, Professor Gibbs published this paper in which they started the sentence by saying, within 10 minutes of starting to eat, a rat stops eating, grooms for a short period of time, then usually sleeps. We define this behavioral sequence as satiety. Its physiological basis is unknown. But what is fascinating here is that if you pay attention, what it's saying is that within 10 minutes of starting to eat, the rat has already stopped eating, right? And they go on to explain that the reason that the rat stops eating is because the gut secretes cholecystokinin. However, the cholecystokinin takes up to 30 minutes to peak in the bloodstream. In fact, it can only be detected after 5 minutes in plasma. But what does it say here? That within 10 minutes, the rat has already put pajamas on and is already sleeping, right? So there is an entire vacuum that within those 10 minutes that we don't understand how it is that the stimuli is encoded at the periphery, transmitted to the central nervous system, and certainly is guiding behavior. And here's the work of Professor Teresa Lieber, that in which she used this labeling methodology. And what you're going to see here, that within 3 seconds, this is a mouse drinking in real time. Within 3 seconds, you have food in the stomach, and in less than 10 seconds, that food is already in the intestine. Let's take a look at that again. So this is impressive, because within 10 seconds of started to eat, the mouse has already gotten food inside of the intestine, and the intestine has already had the chance to figure out what is it that it has been ingested. And as you know, in sensory biology, you know a little bit of a stimuli to make that perception of the world. So that is the famous Umwelt, that we only leave the representation of our senses. So the gut doesn't need to know the entire reality. It only needs a sampling of the reality to be able to know that we ate an apple versus having a rice, for instance. So have you noticed that when we go to a grocery store, somehow we go for milk, but at the end in the cashier, magically we extend our right hand and we grab one of those Kit Kats, and without even realizing after paying, by the time that we have gotten to the parking lot, we have already eaten 2 or 3 of those bars. I am guilty of that. And then at some point, I read this book called Predictably Irrational that brought to me the concept that there are hidden forces that shape our decisions. And it has been known for quite a while, since the 1950s, that there is a reinforcing effect of food entering the intestine. And in 2008, Professor Iván de Araujo showed that even in the absence of taste receptors, animals are capable of distinguishing sugar from water, but not sweetener from water, meaning that sugar is distinguished in the gut. And Professor Tony Sclafani, he gets a lot of the credit because he did quite a bit of behavioral work since the 1980s. And in 2010, he actually suggested that glucose in the intestine will condition a flavor preference. And he went in so far to suggest that perhaps it's sodium glucose transporter 1, the receptor. And in 2010, Professor Charles Zucker at Columbia, he also showed, he further confirmed this hypothesis that the gut is driving this so-called sugar preference. And they have called it sugar preference primarily because of the behavioral studies done early on by Tony and others, in which they had to pair a stimulus because we didn't have the equipment to be able to rapidly discern the choices of the animal. And in this paper, Professor Zucker, he highlights very clear that it will be of interest to determine the cells that are driving this behavior. So as you can imagine, we thought that perhaps these neuropods may be the cells driving the behavior, because that would be very important for understanding our biology, understanding that there is a sensory system in the gut that is constantly driving our choices and perhaps other effects. So we set up, for a while, it took us some time to set up this automated behavioral phenotyping system. And then when we gave the choice to animals of sucrose versus sucralose, then after the animals have tested this for a while, if you expose the animals for like a day or sometimes even less, then very quickly the animals will be able to tell you what it is that they prefer. So animals that have tried sugar, then within seconds, 60 to 90 seconds, they show a preference for the sugar. And like I said, this is in the absence, even in the absence of sweet taste receptors. So then the second thing is optogenetics. As you know, a revolutionary technology that if you turn on hunger neurons, then all of a sudden, the mouse starts to eat, even if it is sated, and if the light goes off, then the mouse stops eating and is thinking like, what did that just happen, right? So since 2014, we began working on how to bring that technology to the gut. And immediately we ran into an issue, which was the fiber that was used for the brain was incompatible for the gut because it's rigid. But Professor Polina Nikieva at MIT, she came to Duke in 2017, and I remember she visited us, and she said, hey, Diego, I have this fiber. Do you think that it will be of any use to you? And of course, we were very excited, and we began working, folks in my team, Dr. Kelly Buchanan, Dr. Laura Ruprecht, and certainly Professor Maya Kelber worked on this project. And immediately, we were able to outfit the mouse with this fiber. So the fiber is not going inside of the brain, it's going underneath the skin inside of the stomach. And then in this way, using optogenetics, either channel rhodopsin or halo rhodopsin, we're able to silence or excite these neuropods. So as you can see here, when we show the control light, the animal is still able to recognize sugar. However, when we silence those neuropods, then the animal literally becomes blind to the sugar. And it becomes blind to the sugar only within these first 60 seconds. And this is in the absence of any effect of pancreas, motility, or any other things. And when you remove the light, the animal is still capable of distinguishing the sugar over the sweetener, meaning that this is an effect like the retinal cone cells in the eye that by detecting the wavelength of light will be able to tell us what is red versus what is blue, right? So likewise, the gut, through the neuropods, by detecting specific molecules, is able to guide our appetitive preferences. And then, as you can imagine, when we silence glutamate and glutamate signaling, then the behavior goes away. And we had demonstrated early on that these cholecystokinin label neuropods, they also release glutamate. And eventually we had published this full paper that I highly recommend for you to go and take a look at it and take a look at the details. So here is a video summary of the whole concept.
Have you ever wondered where our sugar cravings come from? Scientists have long known that our preference for sugar does not depend on its sweet taste alone. Instead, our preference for sugar depends on a sensation arising from the gut. But how the gut guides our preference for sugar over artificial sweeteners was obscure until now. When sugar molecules enter the intestine, they are recognized by sensory cells. One of these is the neuropod cell. Neuropod cells synapse with the vagus nerve to tell the brain about sugars entering the intestine in milliseconds. We wondered if these cells are necessary to discern nutritive sugars from non-caloric sweeteners. We found that neuropod cells sense sweeteners using the sweet taste receptor T1R3. This causes the release of the neurotransmitter ATP. But sugars are sensed differently. Neuropod cells sense sugars using SGLT1, an electrogenic sodium glucose transporter. When glucose enters through SGLT1, neuropod cells release the neurotransmitter glutamate. As such, neuropod cells convey signals from sweeteners using ATP and from sugars using glutamate. But does the animal preference for sugars depend on neuropod cells? To answer this question, we used optogenetics. This technique allows researchers to silence or excite neuropod cells while a mouse is presented with a choice of sugars versus sweeteners. We discovered that when neuropods are silenced, the mouse cannot distinguish the sugar from the sweetener. It becomes blind to sugar. A mouse presented with a bottle containing sweetener consumes only small amounts of the liquid, but exciting their neuropod cells causes the mouse to double its intake of the sweetener. It drinks the sweetener as if it was sugar. By sensing nutrients, neuropod cells convey rapid subliminal sensations to convert food into feelings.
So one of the open questions that we had at the time is like, would these cells be encoding not only the salience of the stimulus, but also the valence of the stimulus, meaning that is this stimulus pleasurable? And what I can tell you, like the next few slides that I'm going to share with you, is that when we stimulate these cells, we are able to cause a real-time place preference, which is a proxy for reward, a good feeling. So animals that feel good in a place, like for instance, in your kitchen, you will always feel at home. Why? Because it is paired with a good stimulus of food. So here you can see here that when we stimulate, this is just quantified, when we stimulate with optogenetics, then the animal prefers that. But in a negative mice, we don't get that effect. The same thing it is if we do intralipid, in the case if we do fats. However, when we silence cholecystokinin, which will be the hormonal aspect of it, we don't get rid of this effect, of this preference for the place. In fact, it seems to be enhancing a little bit that effect, the hormonal effect. But what is fascinating is that if we silence pharmacological, the glutamatergic signaling from these neuropod cells in the proximal small intestine, we get rid of that good reward feeling. And if we go even farther down, and it looks like I have gotten rid of the data, but if we go farther down and we knock out the vesicular glutamate transporter 1 into these CCK cells, we have the same effect as you will see here with the pharmacological blockade. So finally, to wrap up this concept, is that for the last 150 years, we have been focusing up here. And now with all of these technologies, we have to start to look down here inside of us, because it's the junction of the brain with the body that not only brings us that ability of consciousness, but also that the idea of mind, that together we can navigate the world through our own umwelt, but also by intersecting with the umwelt of other people, who will see the world in their own way. And just today, I saw that there were 2 papers that came out on this topic, and you see here that these enterochromaffin cells, these serotonin cells, they seem to be different between crypt and villus in the gut. And this is by Professor David Julius. There is also this other paper by Professor Zucker, a body-brain circuit that regulates inflammatory responses. And as you can see, it wouldn't be surprising, because within our intestinal cavity, there is about 8.5 meters of gut house in the abdominal colon. And very rapidly, I just wanted to leave you with a story that we're working in the laboratory, that we discovered a while ago in 2014, that these PYY neuropods in the colon, they express toll-like receptor 5. And toll-like receptor 5 is fascinating because it's a classical molecule in the immunology field that is known to recognize microbial associated molecular patterns, microbial associated molecular patterns, or MAMPs. And what is very interesting is that when we knock out that TLR5 in these PYY neuropods, a signal from an enema of flagellin into the lumen of the colon does not reach the vagus nerve. And here you see it quantified. So if you put flagellin into the lumen of the colon, you get a very rapid vagal response. But if you get rid of the toll-like receptor, specifically in these PYY cells, the signal, does not reach the vagus nerve. And most fascinatingly is that when you knock out these toll-like receptor 5 in PYY neuropods, the animal becomes obese over time. And it becomes obese because every time that it goes to eat, it eats for a little bit longer, as you can see here, like a couple of minutes more, and a little bit more, like a few 100 milligrams more, which over time add up to obesity. So with these stories, I hope that your umwelt expanded. And when you go out there in the world, you realize that we are truly like living our own world, because it is our senses that helps us to perceive the reality in which we live. However, that is not everything that there is to perceive. And with that, I want to thank my entire team, and I specifically want to highlight the work of Dr. Maya Kelber, who has been a champion in this area over the last 10 years in our team, and also the work of Dr. Laura Ruprecht, who has been working our team also for about 6 years. Quite a bit of the work on the toll-like receptor 5 was of Winston Liu, and now of Nama Reicher and Emily Olwe. So, and finally, as you heard, we have this community called Gastronauts. So if you would like to know more about it, please log in into thinkgastronauts.com. We have a global seminar series every month, and also a global symposium. It's going to happen, the next one, in June of 2025, somewhere here in the United States, and you're all invited to come. So thank you so much.